Okay, so welcome everyone to the last session yeah, I took the liberty to shorten the title a little bit. So this is our panel discussion of Germline database working group about beyond inferred alleles, improving quality, scope, and openness of GLDBs. Probably to start with just a short comment for those of you who are new. The reason why we are so intrigued with the potential of inference of genes is, of course, that in contrast to all other transcript genome relations that we have in biology, we have here this unique or rather unique situation of, especially for immunoglobulins, of course, of somatic hypermutation, but for both Ig and TCR of the situation of exonuclease activity during the rearrangement. And this, as Andrew just alluded on for the final nucleotides, makes inference very difficult and not straightforward because it's really necessary to have people to look at the thresholds that people took when they did this inference. We do have good tools, Tigger, Partis, IG Discover, just to name a few of them. But nevertheless, this is the reason why we think that the establishment of AIRRC has been such a huge step forward. Okay, and but so coming from that one and the things that we achieved over the last 18 months, we would like to now move ahead and look a little bit further into the future on what would be reasonable goals to set out for the next couple of years. And I'm very happy to have these 5 gentlemen. So it's all gentlemen, so because nowadays, of course, the women are now out and slaying the dragons, so we can have a little chit chat. So first of all, Henk-Jan van der Ham from Enpicom is here. So he's what's your title again? Senior scientist at Enpicom, and the company is providing services and products to the pharmaceutical company centered around immunotherapy for cancer. Then William has already been introduced, so he's working at Birkbeck College of University of London. Mike Stubbington from 10x Genomics is working at 10x Genomics, and probably most of you know him still from his previous work, also on single-cell sequencing. Then Martin from Karolinska Institute is the developer of IG Discover, so one of the tools that we're using to do these inferences, and has also a special interest in assembling macaque genomes. And then finally, as our virtual attendant from Kentucky, Corey Watson from University of Louisville, who has been one of the leading people in the mouse analysis, but also has a long track record of doing genetics, both in human and also the macaque. Corey, are you here? Okay, so so we're gonna take you last, but so to open up the discussions, we can probably just move through the row. If each of you could give a brief comment on what your perspective is, how we could move forward.
So I think in terms of what Germline databases should provide, there are sort of 2 main things. One is deep coverage of the rarely commonly used species. So strains, alleles, gene usage, ethnicity in humans and strains in animal species. But we also need to find a way of providing really good solutions for less common species. If there are 3 people in the world interested in the repertoire of some African cichlid, for example, then they should have a way of contributing to a GLDB that's good for them. Another thing that we need to do is to start combining genomic and REP-seq data. So at the moment we have our inferred alleles, we have the genomic data that's going into IMGT. As long-read sequencing comes in, PacBio, you know, that Corey will tell us has its own problems. So we'll get the best results by combining the two and using REP-seq data to validate what we find genomically. Another issue is tailored germline sets for particular populations and particular strains. And I think expanded support for constant and non-coding regions as well will start to become more important. And as we look at differential usage of germline genes, we're likely to find some of the answers in those parts of the sequence space. What we're looking for with this, what we can get with it, cleaner and more accurate characterization of function and response. And in particular, if you're, for example, doing a vaccine study and you want to understand how a particular antibody develops over time, well, you need an accurate lineage tree, and that means you need to know accurately what the germline genes were to start with. I think we'll also, out of the understanding we get better tools for annotation and for inference. And if you look at the inference tools today, they have heuristic features. There are numbers that are set to seemingly sensible values. They make assumptions, and I think more understanding will let us develop those tools and get better inferences. And maybe out of all of this, we get some more progress on correlates of protection, but that's such a hard problem, we'll just have to wait and see. Roadblocks, as we've seen with some of these other species in the GLDB working groups, naming is a big issue. Strains are a big issue. There's work to do on all of those before we can start moving forward. The read length for REP-seq is a real problem. Funding is the final blockage I'll mention. This whole effort needs to be made sustainable, both on the human side and on the infrastructure side. And to do that, we need a sustainable funding model.
Currently, we're seeing a move in pharma and pharmaceutical companies that are trying to use repertoire data to evaluate the efficiency of immunotherapy treatments in cancer. So this is one of the major applications of the somewhat more novel applications, I would say, that you use this as a readout for, say, cancer therapy rather than just studying the Ig genes for the sake of the genes themselves. So there's a lot of companies and corporations interested in using, for instance, the databases and the germline databases that are being curated through these committees. One of the roadblocks, I think, for that is to try and bring in the industry so that they can also contribute to this process of curation, and also to enable the industry to use the databases that are currently available on clear terms. So in terms of licensing and these sort of things, there is a number of issues there that make it difficult for especially smaller companies to use the databases. So that is something I'd like to mention. Yeah.
One of the things that we noticed, one of the things that we noticed when we're developing IG Discover at the beginning was it's not good enough to just try to infer novel alleles. You really have to do it, as has been mentioned earlier on, in the context of the entire genotype. And once you start doing that amongst multiple individuals, you start noticing variability in genotypes, both in terms of copy number variations and also allelic variations. We have embarked on a project where we're looking at different population groups in humans and also in different groups of macaques. And in both cases, we're seeing a lot of variability, which seems to be population specific. So I think once we start getting a more comprehensive database of both V genes, but also D and J genes, where there is also a lot of variability, I think we're going to be able to help be more clear in our analysis of repertoires. Now, an individual repertoire will actually be derived from the V, D, and J alleles from that individual themselves. And many of these particular alleles are not present within the current reference databases. So I think the future work should really be towards defining the V, D, and J alleles which are present, and have means of actually looking at an individual level and a population-specific level of these components.
Thanks. So when we're thinking about using these databases at 10x, we're thinking about how we can use them to help our users interpret their data. So they will generate sequences, but we want to provide the software that goes on top of that to enable them to analyze those sequences, annotate them, and extract knowledge from them. And we really want to do that for all of our customers and users, whether irrespective of their computational sophistication. So we want to make things like loop cell browser and everything else that you can use on a laptop without needing a bioinformatician. And in the VDJ space, that really is underpinned by Germline databases and enabling our data to be assembled and annotated for the users. And so, of course, we need those databases to be good, comprehensive in terms of sequence and annotation of names, but also, I think, going forwards and thinking about extracting even more information in terms of gapping and region coordinates for the CDRs and things like that, and well structured to enable us, when we're thinking about how to use them, to go in and extract the information, and ideally with good, rich programmatic access for us to get at the data in a way that is useful and easy to do sort of automatically and scriptable. Something else that I think is really important is clear versioning, so that we can know and our customers can know what version of the database they're using, and which I think is important when you're publishing, when you're talking about your data. Has the database been updated since you published? And finally, particularly sort of the space that we're in and that we're distributing software and tools, but we are a commercial organization, we need a path through which we can make these databases available to our customers through our tools and through our distribution mechanisms. We can, of course, enable people to build their own references based on things they go out and access themselves, but I'm pretty sure that not many people do that because it's an extra step that's involved in actually getting the data. So if we are able to make those data available to our users so that they can go out and do experiments and understand them in the context of these databases, that's very powerful for us.
Okay, thanks a lot. So I would already start with taking one of Mike's comment and throw it onto William. So one of the things that you said is that you need to have versioning and programmatic access. So I guess the programmatic access will, well, we have an idea on how that works, but so could you allude a little bit more what type of things you would need for clean versioning? And William, could you comment on how you see that in terms of implementation as we currently have it in OverDB?
So I mean, I think I sort of envisage this as you do for genome builds and things like that. So you know if you're building against the ensemble reference, you know which build of the reference and annotation version you're analyzing against and that you've used to analyze, say, your transcriptome data. And I think something like that would be extremely useful in terms of Germline databases. If you look at, say, I mean, a simpler example, GLDB, where you can just look at commit histories in GitHub. And there's a continuum between those two things, I think, where you actually just lock the build and you say this is build number, whatever, or you can refer it back to a commit history. I think either is okay because it is at least an explicable reference point that you can say we did all of our analysis against this version. I'm very interested to hear where you're thinking of sitting on that spectrum.
Well, one of the things I didn't realize before I started on this is that gene names change. And obviously sequences change as well, and that happens more frequently than one might think. So yes, it really is important. I should say we're not supporting any germline sets on OGDB yet. We will, but not yet. But within the structure that I outlined in the last talk, we have a concept whereby we record the curator, which could be a body or it could be an individual, and then we have a versioning number underneath that. And certainly what we envisage for AIRRC-curated germline sets, for example, is that AIRRC would be the publisher, we would publish a clear version number, and there would be a DOI for each version in a public repository somewhere so that you know it lasts for a long time.
Okay. Good. So moving on from versioning to licensing, I think that's something that now from both. Yes, I can try. Sorry. Moving from versioning to licensing, that's apparently something that has been some tripping stone for both industrial people here on the panel. So this is also something that we explored a little bit in OGDB. Right now, so after some consultation, it was decided to go for CC0. So what are the main aspects for versioning? Is it about legal security? Is it about openness? Is it about that you can sub-license? I mean, you can.
So I think it's about, because the way our kind of philosophy with our software is that we want to just send out a bundle of code that can be installed and run very easily by our users, and say Cell Ranger is like that. There's very little dependency installation, and we have found that that has been very enabling for our users to just get going straight away without having to think about getting anything else installed. And that comes down to the references as well. The ideal situation is that, you know, when we ship out a transcriptome reference or a VDJ assembly reference, that those are unencumbered by licensing restrictions, or we have managed to agree licensing terms that enable us to send those out to our users. And I appreciate that that's not trivial in cases where our users may be commercial, span the spectrum from academic to commercial, but I think that would be great for us, is that just to say, we have the best germline database as our reference, because that's going to give everyone the best data and the best insight into their science, but also that can be used as part of our distribution.
Mm-hmm.
Yes, that's indeed also what we run into, that users don't want to be bothered with selecting a database. They just want to get their analysis done. And they typically ask for the services of companies like ours, because they don't want to get into the nitty-gritty of the analysis. They just want to get their result out. Additionally, I think you know, companies are going to start finding their way around this by either curating their own databases, and that means that eventually we could end up with every company having their own sort of version of a germline database, which would certainly not help the field, because everybody would be using a slightly different set of annotated genes. And I think that would hamper progress of the field in general. So I think it's important to at least make sure that we can all tap into a common source of germline genes.
So, but in terms of your business model, would it be possible for you also to contribute to such germline databases? So, for example, if your customers infer alleles, then that you help them to basically, so to facilitate the process of this information getting back to academic databases?
I mean, I think that would depend on our customers, but we'd be very happy to. We're also very happy to contribute to all our internally generated sequencing data, of which there is a fairly large amount, to help in these aims. And I think that two-way process is important.
Yes, exactly. So if customers are indeed willing to or allow their data to be made public, then certainly we can work with that, yes, to contribute them.
Okay, so basically to summarize that, it's mainly about ease of use, legal security from your side, share alike, or incentivizing these things, or having that it's not that you would consider proprietary databases to be really an asset, but rather a problem because it costs you money and in the end it splits the community. Okay. Good. Then to move to a slightly different topic and going more towards the inference again. Martin, one of the comments that you made also about thinking about whole genotypes. Of course, there is an interplay between known alleles and alleles that we infer. So, how much are we moving into some hen-egg problem if we don't have good and easy programmatic access to databases?
Well, one thing we notice once we start looking at a very large number of genotypes is that we can identify a lot of the IMGT alleles in our genotypes, but there are quite a lot of alleles that we never see, and it doesn't really matter which type of library preparation we're using, you know, race, different types of 5' multiplex libraries. So there are quite a lot of alleles that are indicated as functional in the IMGT database that we never see in our data. And once we start accumulating our own inferences as well, then the accumulation of sequences for particular genes allows you to do things like population studies and evolutionary studies of alleles. However, some of the alleles that are present in the reference databases at the moment, they don't quite fit in. They look very different. So we think there is a lot of problematic issues with specific alleles that will cause problems if people don't realize that there may be reasons why you should not use these in population studies. So I think it is an issue that probably needs to be addressed, particularly when we start accumulating very large data sets, because it's going to be impossible to do good evolutionary or population studies if you have a lot of incorrect alleles.
Okay. Corey, are you online now?
I think so. Can you hear me?
Okay, cool. So, I mean, as someone who's worked on human macaque and mouse, is there something that you would like to add to Martin's comments?
Not at the moment. I think that both Martin and William have sort of covered what I would have said in an introduction anyhow. So at the moment, not really. I would say that, you know, the interest that my group has is trying to understand that the extent to what role variation outside of the coding segments has in determining repertoire variation. And so because of that, we're generating a lot of genomic data outside of the coding segments. And I think that's going to be a big topic moving forward, at least in my mind, is trying to decide what value those data have from a database and a data curation standpoint. And I'd be happy to elaborate on that further, but that would be the only thing I would add.
At this point, I'm not the technical person. Then
I assume that's Jamie talking to me, but I can't understand what she's saying.
Ah, okay. Yeah, that's better. That's better.
Okay. She said that she couldn't hear me. Okay, perfect. I'll be quiet until you ask me another question.
Yeah, well, of course. Okay, so now we know how this one works. Okay, so but Corey, just to keep you on the hook here, in thinking about allele frequencies and populations, I mean, it should be probably relatively easy to annotate these things technically on the database. But where do you see the main challenges in accumulating data sets that would allow us to do this?
Yeah, so I mean, if we're talking about coding variation, you know, I think as Martin and William both already pointed out, you know, that's sort of happening by default with a lot of the work that AIRRC and OGRDB are doing, because you, in some respects, get that for free from rep-seq data. The challenges have been historically for non-coding variation technical, in the sense that, you know, there haven't been very robust ways to assay, especially in a high throughput way, variants that, you know, occur outside of the coding segments and intergenic space. And there's been an underappreciation of the potential role of those variants, I think, historically. But I think all the data that we're seeing come up now is basically showing that a lot of those variants actually probably have pretty important roles in the variation that we see in repertoires. And so I think a challenge moving forward is just going to be, one, implementing genomic tools that allow individuals to go into a cohort of interest and actually accurately sequence those samples and accurately call genotypes in those samples. And then that's going to have to be done in a way, obviously, where you can begin attaching some sort of functional information to those variants. And the simplest way I would imagine, which we're already starting to do, is just ask questions like, if I take a SNP, any given SNP in the IGH locus, does it correlate or associate with a functional feature that we see in the repertoire, like something like gene usage, to make it simple? And the answer to that question is yes, we do see lots of examples like that. And so I think something that we have to think about moving forward is there a place for that type of information within, say, a GLDB? Can we store that type of information, and can it be leveraged for people who are analyzing rep-seq in any context?
Okay, thanks. So Henk-Jan, just to move that directly over to you, is there, would you see any application for your customers for this type of information?
I think that strongly depends on the customer. So, I mean, there are customers with varying different projects. There are enormous amount of applications of repertoire sequencing, so it would really depend on the customer, I think. Yeah, so I think it's difficult to say upfront.
Okay. Okay, so then switching gears a little bit again. As some of you know, we, so I think at the last meeting we introduced the terms of the AIR data commons, which is the idea with that the AIR community as a whole should work towards linking together or unifying databases, so that this is actually one resource that we all build, maintain, and can draw from. I mean, until now, we, of course, the main player in the field has been IMGT, who has done really magnificent work for over 20 years now. So as we already seen with the work that William has done with OGRDB and the question for programmatic access, so an API, which would be, of course, the foundational stone for if we're trying to federate these things. Where do pretty much all of you would see the pros and cons, or the advantages, or also the risks of going for something like this?
Well, I think the place that it might impact users most is if users could specify the germline set that they wish to use for an annotation. And that could be because they want to use a tailored reference set for a particular purpose. For example, they really want to use one where they know that all the genes have been observed in a population. Or it could be simply that they want to keep the germline set version constant over the range of their analysis. And their analysis might last for a year or longer. Now, I think we could construct an API that would let people pick a germline set, either select one or make one themselves from genes they want to be in there, and reference it, and then pull that into other tools via an API. I think that would be a good thing to do, and perhaps the thing that gives immediate user benefit.
Yes, I think programmatic access would be very useful also for platform tools and all this sort of thing. I think it would be also very easy to then automate pulling in new versions and to make sure that things are up to date and that users can indeed have the freedom to select the specific version that they want, so that they can keep the version fixed if that's so required, and that it's very easy to update and doesn't require a lot of manual curation, intervention, or actions. And that would very much help, I think.
As I'm particularly in favor of users being able to define the gene set that they're using for analysis. Just to switch to rhesus for a second, we originally developed our tool because the rhesus database was actually so constrained. And when we developed the tool, we actually noticed that there was a huge amount of variation between different animals. So it was almost essential that you actually did an individualized database per animal. You don't get that amount of variation in humans, but I think the same principle does apply, that using an individualized set is going to be better. So to be able to select which specific sequences you're using in the database from a larger data set or database is recommended.
Yeah, I don't have a great deal more to add other than I think we've seen over the course of other scientific databases that good quality APIs massively increase their value to the community and enable people to use them in novel ways and combine them to find new things. So I'm always supportive of programmatic access to data.
Corey?
You're here.
Okay. Also, the question to you: Do you think that distributing databases is a reasonable thing to do or not? So Germline databases.
Do I think it's a reasonable thing to do?
Yes.
Yeah, I mean, I might be missing the question. I thought we've already sort of covered this ground, but yeah, I think that makes a lot of sense, and I think that it's going to contribute to ensuring that we have better consistency across studies, making sure individuals are analyzing the same data sets and utilizing data sets in the same way to the extent possible. Did I miss the question? Sorry.
So it was specifically about, do we need a federated system of Germline databases, not necessarily of AIRR-seq databases? But if that was your answer, then of course.
Yeah, I suppose my answer would be that I could see that being useful if we can't wrangle and if we can't ensure that everyone's going to submit their data and to a central place, then yes, I think that's absolutely critical. I think it's absolutely critical.
Okay, so I just got some hand waving that we slowly need to round this up. So of course we come back to the most critical point at the very last, namely how do we finance this? So William, you already alluded on this, and there's probably also a question of the towards the whole audience. So luckily until now, IMGT has always managed to acquire their own funds, but as we're now seeing how much work AIRRC has put in this, we think that it's probably necessary to put much more people power into this process if we really want to make it clean and sustainable. So what would be the ideas on how there would be financing? I mean, of course, we should all be open to going for things like FP9 / Horizons Europe, which have a very clear open data agenda. So we should probably take them and say that this is something that the community needs and it needs to be financed. Would you see any other possibilities for this or any other models?
So there are also quite specific grants that stimulate private-public partnerships, so companies working with academia. I think this might be a good opportunity for a grant like that to be used for this sort of activities, because it's both industry and academia that have an interest and that profit from this type of exercises. So I think that could be quite a good opportunity as well, in addition to the ones you already mentioned.
Mm-hmm.
You could also imagine organizations like Elixir potentially being interested in things like this, but I don't feel like I know the funding landscape well enough to... Oh, sorry, I was suggesting potentially organizations like Elixir who have a strong sort of pan-European data integration openness agenda, but I don't feel like I know the funding landscape well enough to make any very strong suggestions.
Of which, best to my knowledge, Elixir, I think IMGT is already part of that. So, okay, so then last question, William, how often do we have to clone you to get the work done? Just to have a number that we can put into the...
Cloning's expensive. So, I mean, the only wild idea I'll throw in, and it really is a wild idea, is you know, we could look to set up some kind of royalty basis for funding this, under which perhaps academia pays on some estimate of what its royalty contribution would be through grants, and industry could pay more directly. But I think we need to do, we need quite a few discussions, because as I understand it, IMGT work is funded in various ways at the moment, and we'd need to understand and unpick that. And we need to come back and look at the AIR philosophy, which has been that all of us feel the work that we're doing is for the public good and should be available for free for the public. You know, is that something that we stand by so much that we refuse to take any money for it? I think that's all from me, really.
Okay.
Can I add that I think to me the biggest issue is going to be longevity. I mean, you know, eventually William won't want to do this anymore, and so I think, you know, we want to be careful that OrgDB doesn't go away, and I don't think that that's the case. But you know, I think that we should be thinking about how do we integrate what William is doing into existing established systems that kind of we expect to have more longevity by default. I'm thinking like EBI or NCBI, and I'm saying this not really knowing how that would work, but just thinking about it in a very sort of superficial way. It seems to me like, you know, we can get money to sort of get the ball rolling initially and to make this thing real, but I feel like, you know, my concerns would be how do you make it last for a long time, and that's one way that I would see potentially doing that, especially given that we have connections with our other efforts, AIR efforts with groups like NCBI and EBI and whatnot.
Okay, so basically you now ruined my last words, so thank you for that. But nevertheless,
I aim to please.
No problem. Okay, so we really have to close up now. So thanks to all the panelists for sharing your insights. Thanks to all of you, and please keep discussing these issues. We have 3 people from IMGT here. Please engage them. We might not have always agreed with them, but it's really necessary to talk to each other to see what we can do together as a community. And we have to realize that this is really one of the most central community resources that we have and that we have to build and maintain. All right, so otherwise see you tomorrow.
Yes.